we provide some quantitative description on the both response by intensity. Here is different intensity of ultrasound, and PRF ranges from 40 Hz to 200 Hz. This brain functions activity change with the intensity of this stimulation. After the stimulation, and we actually see that the T2 weighted MR imaging shows there's no obvious damage for the brain tissue. And also the 30 days after the stimulation, the behavior of the monkey does not show any kind of abnormality. Ultrasound stimulation of the primary visual cortex enhances the functional connection streams on the target and the visual neural network, as well as the cortex related to visual information process. This is quite exciting because, for this kind of non-invasive tool, it could be used to investigate the core in the neuron circuit connection and to get out the connector for many unknown of these neuron circuits. Actually, in another study, we tried to search for the abnormal circuits and the function areas in the brain network connections, and to try to screen out the precise stimulation targets for this system, for this modulation. For this kind of connection or knowledge is very important for the brain disease study. For depression, for Parkinson's disease, for this, especially for the disease with very complex kind of brain dysfunction and complex connections. This time we tried the dorsolateral prefrontal kind of cortex. We construct the picture, the image about the MRI image, and the target location and treatment planning we carry out using the MR Alpha kind of software we developed. We figure out the brain zones we want to stimulate and navigate this stimulation using the software. The transducer frequency is 300 kHz and the amplitude is around 1 megapascal. Its PIF is 100 kHz, 100 Hz. The dorsolateral prefrontal cortex, DLPFC, this nuclear target. And from this MR image, we can build signal. We can see that kind of stimulation. The DLPFC has evoked functional activation, positive or negative of the target, and nodes in the associated of the neuron circuits, not only on the target but also on this kind of associated neuron circuit. The signal are obvious. So this is a very interesting result to help understand these neuron circuits. This stimulation causes a change in the connection streams of the brain network associated with the target, which means that this non-invasive tool has guidance significance for understanding its complex brain function connection, and also especially for the physical intervention and in the treatment of the dysfunction of the brain. For many kind of functional brain disease, this non-invasive tool may play very useful kind of role in the therapy. Well, we come to the conclusion. I think from our study and many ongoing studies, you can notice that this brain stimulation, non-invasive, is very attractive, very fascinating method of developing very safe, effective brain stimulation tool are still a challenge. The underlying mechanism for the neural stimulation, we already have many data to support the mechanosensitive induced ion channel change and induced the neural activity, but some other mechanisms still kind of possible. A series of the brain stimulation tools, including the chip, the portable wearable, the small devices, and the larger planar array for the large animal human being study we developed, It's compatible very well with MRI, as MRI could provide precise localization in the imaging, the brain function induced, the bird signal acquisition, and the temperature monitoring. I think this information is quite important for the next step kind of brain disease therapy study, and also a large animal study to understand the different kind of disease mechanism for the exploring the application for the ultrasound neural stimulation. So I would like to acknowledge many team members of this study, my colleagues, my PhD students, postdocs, the biomedical imaging team at SIT Shenzhen, China. Thank you very much for your attention.